Good day. This is Miguel Alfonso B. Eustaquio, and I am going to report about the production of lipase by immobilized cells of Aspergillus niger. So to start, we are going to talk about lipase. So what is lipase? Lipases catalyze the hydrolysis of fats to produce monoglycerides, diglycerides, free fatty acids, and glycerol. So these reactions are reversible so that lipases also catalyze the formation of acylglycerols from glycerol and fatty acids. So lipases also possess characteristic properties like substrate specificity, stereospecificity, and the ability to catalyze heterogeneous reactions at the interface of water-soluble and water-insoluble systems. So lipase has the ability to be applied in chemo, regio, and enantioselective hydrolysis reactions. Because of that, they have been used in a wide range of industries and applications: fat and oleochemical industries, detergent, production of biodegradable polymers, textile, pharmaceutical, cosmetics, and other than that, they are used in peptide synthesis, biosurfactant production, and resolution of racemic mixtures to produce optically active compounds. Lipases occur in animals, plants, and microorganisms, with filamentous fungi being the most reported species to produce lipase. So microbial lipases show a broad spectrum of industrial application due to their greater stability, substrate specificity, and lower production cost. Microbial products are usually obtained from free or immobilized cells. Immobilization of cells can be considered as physical confinement or localization of microbial cells. So the utilization of immobilized cells as industrial catalyst in continuous processes can be advantageous compared with batch fermentation procedures. Immobilized whole cell technology has several advantages over enzyme immobilization. This paper deals with the immobilization of an indigenous Aspergillus niger mutant for the production of lipase using different entrapment techniques with matrices sodium alginate, kappa-carrageenan, and polyacrylamide gel. The effect of different incubation periods, optimum concentration of sodium alginate, and the reusability of immobilized cells for lipase production were investigated. Here is a quick overview of the methodology used in this paper. So they started with microorganism isolation and then prepared a spore suspension. After preparing a spore suspension, they prepared the immobilized cells using different entrapment techniques. They carried on with fermentation, and after fermenting, they studied the optimization of conditions and the lipase activity. For the isolation of the microorganism, Aspergillus niger AU15 strain was isolated and was improved for lipase production by mutagenesis. So they then labeled the improved mutant as ANT90. Spore suspension preparation started with the organism being subcultured on potato dextrose agar medium. The spore crop of each slant were broken off spore chains to make sure uniform suspension will be made and then filtered to make a 5 times 10 raised to the 6th power spores per ml suspension. The spore suspension was then used for preparing the beads. Immobilized cells were prepared using different entrapment methods. The first method is sodium alginate entrapment method, wherein the spores were immobilized in calcium alginate. Sodium alginate and spore suspension were mixed into a slurry and then dropwise added to calcium chloride. The beads formed were then cured in the refrigerator overnight. The next method is the kappa-carrageenan entrapment method. The kappa-carrageenan was mixed with spore suspension and then poured to Petri plates. After solidifying, they were cut into uniform blocks and then cured in potassium chloride inside the refrigerator overnight. For the last method, the polyacrylamide gel entrapment method, chilled cell suspension and potassium phosphate buffer were mixed and poured in a Petri plate. The acrylamide gel was cut into equal-sized cubes after polymerization and then cured in sodium phosphate buffer in the refrigerator overnight. So the immobilized cells that were prepared using three methods were added to Erlenmeyer flasks with growth medium. So the growth medium is composed of glucose, peptone, sodium chloride, yeast extract, and was kept at a pH of 6. They were then incubated in a rotary shaker for 72 hours. After 72 hours, the growth medium was decanted and the beads were washed with sterilized water and transferred to another Erlenmeyer flask with a production medium consisting of dextrose, peptone, yeast extract, ammonium sulfate, Epsom salt, ferrous sulfate heptahydrate, and sodium chloride at a pH of 6.5. The flasks were incubated in a rotary shaker for 120 hours, and samples were withdrawn at 24-hour intervals and assayed for lipase activity. Lipase production was also carried out for Aspergillus niger-free cells using the same production and medium. For optimization, the effect of different incubation periods, optimum concentration of sodium alginate, and the reusability of immobilized cells for lipase production were investigated. After fermentation, the lipase activity was determined by titrimetry using olive oil as its substrate. So 1 unit of lipase activity is defined as the amount of enzyme required to release 1 micromole of fatty acid under standard assay conditions. The experimental results of the lipase production with free and immobilized cells using various entrapment techniques are shown here in Table 1. So we can see that the maximal production of lipase by free and immobilized cells was obtained at 96 hours. So the present results are consistent with this finding. For Table 2, the immobilized cells with sodium alginate, kappa-carrageenan, and polyacrylamide matrices were used for repeated batch fermentations. So you can see that the productivity of lipase by cells entrapped in calcium alginate was higher than that of the cells entrapped with kappa-carrageenan and polyacrylamide gel. The low yields with kappa-carrageenan and polyacrylamide blocks may be due to diffusional resistances of nutrients and oxygen into the matrices. The beads prepared from sodium alginate were stable up to 3 cycles, and that the immobilization blocks prepared with kappa-carrageenan were not able to retain their shape during repeated batch fermentation and disintegrated after 3 cycles. The blocks prepared with acrylamide were relatively stable, but disintegrated after 3 cycles. Therefore, alginate was considered to be the best matrix for the production of lipase. As such, cells entrapped by sodium alginate were used for further optimization studies. Table 3 shows the different sodium alginate concentrations that were used for the preparation of beads and were studied for the effects on the production of lipase. So the production of lipase was seen to be maximal at 3% alginate in comparison with the other concentrations used. Although 4% alginate gave more stable beads, the yield of lipase were lower. The decrease of production of lipase with 4% alginate could be due to the diffusional resistance offered by the beads. So based on this, the result with 3% alginate was selected for subsequent studies. In Table 4, the effect of curing time on the stability of beads to produce lipase was studied. So the beads cured for 60 minutes were stable and resulted in better lipase production. The beads cured for 30 minutes were gradually disintegrated from second batch onwards, while the beads cured for 120 minutes were more stable but yield at less lipase. As such, 60 minutes was selected as the optimal curing time for subsequent studies. Lastly, the effects of different concentrations of olive oil was studied on the production of lipase using free and alginate immobilized cells. So all in all, the maximal lipase production was observed at 1% olive oil for both cases. This indicated that olive oil is a lipase inducer, and previous studies have already reported an increase in lipase activity when olive oil was incorporated into the production medium. Based on the previous results, it can be concluded that alginate was a good matrix for the cell immobilization for the production of lipase, and that olive oil is a good lipase inducer.